which takes place monthly. We always have one keynote speaker and one early career speaker. The keynote speaker today is Professor Sai Reddy from ETH Zurich, and he is an associate professor and also, among other things, the vice director or co-director of the Bordner Research Center for Child Health. And Sai will talk today about predictive profiling of SARS-CoV-2 variants by deep mutational learning. And after that, we will hear from Sebastian Valkiers from the University of Antwerp on large-scale PCR clustering. So with that, I give the floor to Sai. Thanks so much for being with us.
Thanks, Victor, and thanks to everybody for joining. Yeah, so today I'm going to talk to you a little bit outside of maybe the conventional repertoire type of presentation, although I've done a lot of repertoire research in the past. This is a little bit different, but I think in some ways the principles and the ideas are still somewhat relevant and probably very interesting, at least to this community of researchers. So if you're like me, you're probably feeling optimistic that we've gotten through a lot of the perhaps worst of the COVID-19 pandemic. But you may also sometimes be a bit worried because thanks to vaccination, we are where we are, but we made a lot of progress, but it hasn't been without its setbacks. And if there's anything that is most concerning, it's very much obviously the evolution of the coronavirus. And if you take a look at some headlines just from this summer alone, there's always something that could kind of get your attention and alarm you. Whether it is the new Delta variant when it emerged, we saw how much of an impact it made, but of course there's other variants like Lambda and Delta Plus. And then there was a highly mutated variant out of South Africa. Then there was the Mu variant and some guy talking about COVID-22, and then some new variant called B11523 that's capable of escaping immune responses. And so when you see these headlines, obviously it's easy to potentially dismiss them as fear-mongering, but at the same time, what we've learned from and how we've seen the experience of the Delta variant and how it changed the trajectory of the pandemic should make us cautious around how to think about any new variant that emerges. So the one thing, though, whether those will truly impact the course of the pandemic or not is questionable. But I think we can be 100% confident that the SARS-CoV-2 virus will continue to evolve and new variants will emerge. And as this phylogeny indicates here, if you start from the original founder strain from 2019 in December, you can see the continual pathway of evolutionary lineages that have emerged, certain variants and their subvariants and lineages that come out of it. And of course, notably, variants like alpha, delta, gamma, and beta have all been impactful. Now, looking more closely at SARS-CoV-2, I think that most of you are probably aware that it is the spike protein that plays a pretty important role. It's of course the component that are used in most vaccinations, and it's mutations to the spike protein that are indicated and highlighted in this image that usually characterizes a lot of variants. And when you look closely at the spike protein, pictured here now in gray, this gray structure, and then this darker gray indicates the receptor binding domain. And as its name indicates, the RBD directly interfaces with the ACE2 receptor, which is of course the host 2 receptor that SARS-CoV-2 utilizes to enter into host cells. It's been pretty apparent since very early days of studying the virus that neutralizing antibodies can be used to essentially inhibit SARS-CoV-2 or entry into cells, and the neutralizing antibodies that are typically the most potent bind directly to the RBD, and they bind directly to epitopes that basically block and inhibit binding to ACE2. So what's pictured on the right side here are 4 clinically approved, or at least clinically used, therapeutic antibodies. On the left side are 2 Regeneron antibodies, and on the right side are the 2 Eli Lilly antibodies. And you can see that they bind to different configurations and epitopes of the RBD, but essentially do engage this green part, which is where the ACE2 interface would be. Now, a lot of really good structural biology has been performed on a lot of different antibodies from isolated from various donors, typically infected or convalescent individuals. And this is a really nice classification system that was established by Chris Barnes from Pamela Bjorkman's group. And this was kind of established last summer, more or less. And what they essentially describe is that there seem to be a lot of antibodies that can be grouped together based on how they their structural footprint onto the RBD. The RBD now is pictured in gray. And so class 1 antibodies, in which Regeneron, one of the therapeutic antibodies from Regeneron is here. CB6 is another name for one of the Eli Lilly antibodies. I believe it's LiCov16. And you can see they form class 1 antibodies, which are, characteristically represented by this green structure. Class 2 antibodies, which also consist of engaging directly with the ACE2 interface, are shown here. And you can see that they kind of overlap a little bit with the epitope of class 1. Class 3 is a little bit more distant. What you can see here is Regeneron 10987, which is one of the therapeutic antibodies, and also S309, that is the antibody approved from Vir in partnership with, I think, GSK. So that's also a clinically used antibody. It, however, is a little bit more distant, and it doesn't directly inhibit the ACE2 site. It more or less engages and through some steric hindrance is able to reduce binding to ACE2. Class 4 antibodies are very interesting in that they bind to a very highly conserved site of the RBD. In fact, it's so conserved that the example shown here, CR3022, binds both and neutralizes both SARS-CoV-2 and SARS-CoV-1, the original SARS-CoV virus from the early 2000s. In fact, it was isolated initially from studies with SARS-CoV-1. However, because it's so far away from the ACE2 binding site, the neutralization potencies of class 4 antibodies are extremely weak, and effectively no real therapeutics are being pursued with class 4 antibodies. Class 3, so basically class 1, 2, and 3 are where you can get some potent neutralization, class 1 and 2 being the strongest. And this is essentially where antibody responses, therapeutic antibodies have been focused towards. Now, an important thing to just consider is that these aren't obviously, we're in the repertoire community, so you know the high diversity of antibodies that exist and that the human population is making vastly diverse antibodies, or at least antibody sequences. But I think what this data has started to sort of indicate is there's quite a bit of convergence in essentially the structural conformations, perhaps also some sequence convergence, which is observed in some class 2 antibodies, often sharing very similar germlines. But essentially, it's that there's a structural convergence that means that perhaps these antibodies that fall into these classes could be in some ways partially representative of population immunity. at least immunity of neutralizing antibodies. And I think one of the really interesting indirect pieces of evidence here is that if you take a look at the RBD domain, and these are, of course, the most notable variants classified here, you're looking at mutations, and what we start to see is probably one of the most interesting mutations is in position 484. So E484K has been observed in both beta and gamma variants, and then a host of other variants. And if you initially, beta and gamma variants were originated from South Africa and Brazil. And they actually were, of course, independently, they presumably independently evolved. They also share N501Y, and they also share similar mutations in position 417. So what that might indicate is, and after doing some more extensive studies, it's actually very clear that E484K drives a lot of escape to class 1 or class 2 antibodies. In fact, one of the Eli Lilly antibodies is completely loses binding when E484K is present. Class 1 antibodies also partially are unable to neutralize K417T or N. So this seems to indicate, even though these therapeutic or these monoclonal antibodies are not what all of the population is making, it suggests if there's convergent evolution of the RBD towards particular escape mutations, one might assume there's also convergence in the human immune response, if indeed we believe that antibody escape is one of the selective pressures for variant emergence, which I think there's many good reasons to believe that. Okay, so let me go to the next slide. So variants with resistance basically are, whether it's resistance to a single antibody or groups of antibodies is almost assured over time. So if we look at the blue curve here, we're just indicating that there will be more and more variants over time. I think that's kind of a given with the endemic nature of the virus and the global spread. And if you look at one of the most interesting examples of this is that antibody I was alluding to, LICO-555 from Eli Lilly. It was approved historically last November 2020. It was the fastest ever therapeutic antibody approved, and it was the first therapeutic for COVID-19 approved. Well, by April 2021, so perhaps less than 6 months later, the FDA had revoked the approval of this drug. Why? Because it completely is ineffective against E484K mutations or variants, which had become the more one of the most common class of mutations found in the United States at that time. And you can certainly be confident that not enough people were getting this drug, that the drug caused the evolution of this escape variant. It's most likely it was human immunity that drove this escape. It's just that this drug looks a lot like human immune responses. And then the other thing you can take a look here is a recent study that characterized the neutralization using an in vitro assay with serum. So this is post-vaccination serum 1 week after the second dose of the Pfizer-BioNTech vaccine. And you could see that there is quite a bit of strong neutralization to the original or very similar spike protein. But some of these variants, like very notably mu, but also beta, show a substantially weakened neutralization percentage. So how that could manifest itself is that this could be that such variants would have a higher chance at breakthrough infection, potentially including symptomatic disease and maybe even severe disease. So at some point, if we're only relying on antibodies that were induced against the original SARS-CoV-2, spike protein, like our vaccines are, and maybe if we had exposure in previous years, then we can be assured that those antibodies will eventually lose efficacy to some novel variants. And that is, of course, the biggest challenge in that could very much alter the trajectory of the pandemic, may even prolong the pandemic. And I think we've seen some indications that of course has occurred to some extent already by the fact that the surge of infections that occurred this summer in certain parts of the world. Okay, now, if you've been following this field at all, you may know, or you should know of the work from Jesse Bloom from the Fred Hutchinson Cancer Center. And he essentially developed and applied this method of deep mutational scanning, which is, I'll explain it here in a minute. So essentially, you express the receptor binding domain on the surface of yeast cells, and then you make mutations to the RBD. And the way the mutations are made are single position mutations are made at every position in the RBD tiled across, and the RBD is about 200 amino acids. So you basically have 200 positions times 20 variants. So it's about a 4,000 mutant library. And very important to note, it's one mutation at a time. So there's never more than one mutation because it's tiled across at a single site approach. And then flow cytometry is used, and either the RBD is labeled with a soluble version of the ACE2 receptor, or it's labeled, as this flow cytometry graph shows, with an antibody, like one of those neutralizing antibodies. And then the fraction of variants that have less binding are sorted, and then deep sequencing is performed on this population. And what this allows you to do is essentially identify enrichment in the population of sequences that have mutations in a certain position. Here's a really good example where the E48484 mutation is in this escape at high levels for this LICO 555. That's the bamlanimab antibody that the FDA had to revoke. And you can see in here, E484K is represented. Similarly, you can plot these mutations here in this kind of approach. And if you're, it should not go without notice that this technique has been highly impactful for the field, as you can see by this extremely prolific publication record from Jesse Bloom in the last year alone. Okay, so the problem, however, is that SARS coronavirus 2 is not mutating, or variants are not emerging one mutation at a time. In fact, you can see with this lineage tree, the more distant things are getting. If you take a look, one of the best examples of it is this new variant called C1.2 that was identified in, I believe, South Africa. And it has already 4 mutations alone in the RBD domain. So essentially, we also know that, of course, the beta and gamma variants also have 3 mutations. And so the point is that we need to actually think a little bit beyond single mutations. In fact, if we really want to understand the impact of mutations, we need to start considering combinatorial mutations in the RBD and how these can impact antibody escape. Okay, so what we did is develop this method that we call deep mutational learning. And the idea behind this is we took the RBD domain and we identified regions that have been found to mutate extensively in the viral genome sequencing data in the GSED database, as well as regions that we know are interfacing with the ACE2. So sometimes this sub-region of the RBD that directly engages ACE2 is called the receptor binding motif, or the RBM. And so we took a region of the RBM 484 to 505 that we just labeled for now RBM2. And we also took positions 417 and 439, which have been noted to also mutate in circulating variants. And so we created combinatorial libraries in those regions, and I'll go in a lot more detail in a few minutes. But essentially, we created those libraries, and similarly to Jesse Bloom, we expressed them on the surface of yeast, and then we performed we performed flow cytometry to first select for variants that bind ACE2 and then escape binding to ACE2. And then all of the ACE2 binding fraction, so these are really the only variants that would ever naturally emerge, most likely. They would have to maintain binding to ACE2. So we sorted the libraries, and then we come with the ACE2 population and we label them with one of these 4 clinically approved antibodies, the 2 Eli Lilly or the 2 Regeneron ones, which represent 3 those class 1, 2, and 3 antibodies that are likely the most relevant for neutralization. And so those are labeled individually, and again, variants or mutations that in the library that maintain binding or escape binding are selected. And then all populations are then prepared and used for deep sequencing. And so we generate this large deep sequencing data set of binding and non-binding for ACE2 and for antibody binding and antibody escape. And then we train machine learning models that are able to predict binding or escape. And the reason this becomes essential is that the protein sequence space is so large that our experimental libraries would never cover the combinatorial mutational space. But that is where machine learning can actually come in and address this problem. So I'll walk you through the whole pipeline, as well as a lot of the interesting data that's emerged. Okay, so the first thing that was essential is the design of the combinatorial library. So what we did is we used the data from Jesse Bloom's original publication, where he performed DMS on ACE2 binding. That's a Cell paper from 2020. And we used it to essentially identify within this region of the RBM, 484 to 505, positions that could tolerate mutations based on DMS, and then as well as positions that could not tolerate mutations. And so some of these positions remain fixed because we know from the DMS data that if you make a mutation, for example, in position 486, either the RBD just cannot express anymore or it has no ability to bind to ACE2. So we figured that it's better to be conservative in those areas rather than putting mutations, which of course would de-enrich your library for any kind of binding or expression. Altogether, this library from 484 to 505 has approximately a theoretical diversity of 6 times 10 to the 12. So just for perspective, yeast display libraries could only typically get to the highest of about 10 to the 9. However, in a more routine academic setting, it's probably 10 to the 8 at best. So you're still 4 orders of magnitude lower than the possible diversity, which is why experimentally, if you did this experimentally alone, you would not really capture a good representation of the sequence space. Another thing which will be critical to understand in a little bit, especially on the machine learning side, is to actually make a low distance mutagenesis library. So what that means is that we made something called the tiling mutagenesis library. And so we went through this 484 to 505 region, and we basically added 3 NNK codons. So NNK means any of the 20 amino acids can be encoded. So only 3 out of the 14 positions that we vary, because remember, we keep some positions fixed no matter what. So of the variable positions, 3 out of the 14 are introduced with mutations, and then they're tiled across. So this actually turns into about 220 different oligonucleotides that you order and then assemble your library. The library is relatively small, and you can completely interrogate this by yeast display because it's only a little bit over 1 million. But what you'll see is that this data from this library becomes essential, incredibly powerful when it comes to machine learning, because you actually need this low distance information. Okay, so here's just a quick look at what the yeast display and deep sequencing looks like. And so these are, sorry, I should mention there's also the 2CE library. The 2CE library is positions 484 to 505, plus position 417 and 439. Remember, 417 is found in a lot of natural variants, and it does directly engage with ACE2, and 439 is also found mutated in a lot of circulating variants. And there we just put NNK codons. And so all of these libraries were then screened experimentally here by flow cytometry. And something I want to point out is that the gates that we draw are clear binding populations or clear non-binding populations. Pictured here is ACE2. binding. And the reason for that is that essentially there is this large intermediate space, but we think that for classification purposes, as well as for potentially just training models that are more or less trying to be classifiers, it's better to not take this intermediate population because it's very noisy and there would be some challenges in trying to learn from that. So this creates very clean data sets for learning. And that's different than Jesse Bloom's approach, where he sorts, their group sorts for even reduced binding and then just looks at enrichment scores. So there is some nuance to the experimental protocol. And if you just take a look at these sequence logos, this is after the deep sequencing, the binding and non-binding fractions look relatively similar. Now, of course, there's a few different new amino acids that go up or down in certain positions, but you certainly could never suggest that you could look at this by eye and identify what a binding sequence is and a non-binding sequence is. And that's, of course, where the beauty of machine learning will come into play, that it will be able to identify the high dimensional patterns that are buried here that separate these two populations. Okay, so of course we did the same for the 4 therapeutic antibodies, but this is, remember, with only the ACE2 binding fraction. The point is you don't really need to predict antibody escape if an RBD doesn't bind to ACE2 anymore, because it's probably irrelevant. It probably will never emerge in the viral population. So we only focus on the ACE2 positive fraction. And then again, we sort for clear binding or clear escape variants. And once again, I'm just showing the 2 CE sequence logos here, but you can see for each of the therapeutic antibodies, the binding and non-binding fractions look relatively similar. Okay, so once we've generated all of this deep sequencing data, then we can start to make machine learning models. And so what does a machine learning model exactly do here? So you take these sequencing data, and I'm skipping the fact that we did a whole lot of pre-processing and balancing of the data sets, and there was quite a bit of work to figure that out. So it was it's not as simple as it's pictured here, but for the sake of time, I've simplified it. So these fractions of sequences are then encoded. So you have to turn them into an input matrix so that you can input them into machine learning workflows. Because we have deep sequencing data, we just use one-hot encoding, which is just essentially turning each amino acid into a row in a vector this way, instead of using any kind of other biophysical features or other encoding schemes. And then we essentially train different machine learning models. What's pictured here is a random forest and long short-term memory recurrent neural network, or LSTM-RNN. Those are pictures because those are the final models that we've selected, which I'll show you why in a minute. But once we've trained these models, essentially it's supervised machine learning. So the model is learning what sequences from the training data, what sequences are associated with binding and what sequences are associated with non-binding or escape. And then once those models are trained, of course, you test it on some test data, and you can determine what the output is will be a probability. So the final output is that every given sequence that you put into the model will predict value, either the RNN or the random forest, a probability that it binds to ACE2, or a probability that it binds to an antibody. And if for the antibody, just so it doesn't get too confusing, the lower the probability, the more likely it is to escape the antibody. For ACE2, the higher the probability, the more likely it is to bind ACE2, okay? And so those are the outputs, and then you can do different approaches, and we ended up doing rather simple consensus model where both models agree. We typically have high confidence that the prediction is correct. And there's different thresholds and things that I won't get into right now. So that's literally what happens is you're training models so that you could take any given RBD sequence and get a probability that that sequence binds ACE2 or that sequence escapes a given antibody. Okay, so when we benchmarked the machine learning models, we first used the ACE2 data. And what's pictured here are a class of very common models that are used. Some of them are pretty simple, and they were trained using default parameters of scikit-learn. So basically, k-nearest neighbors, logistic regression, naive Bayes, random forest, RNN, SVM with a linear kernel, and SVM with an RBF kernel. And you can see that hopefully, this is just the accuracy scores. We also have precision scores here at the bottom, that RNN and RFs are pretty good. SVMs are also approaching it. All models are actually pretty good overall. But the RNNs and RFs are a little bit better. The SVM could argue is better than both, but it's generally a lot slower to run. So ultimately, that's why we chose RF and RNN as our standard models to move forward with. Now, a really important thing, so everything else for the rest of the talk will be RNNs and RFs, is that really understand your training data. So our training data is both low distance and high distance. Okay, so what that means is if you look at the library 2C and 2CE, remember it was a combinatorial mutagenesis library where up to 14 positions could be mutated, 12 or 14 for 2C and 2CE. Library 2T is a low distance library where only 3 positions were mutated at any one time. So if you look at here, the maximum diversity, these are different, but more importantly, the distance from the original Wuhan RBD sequence is quite low or is quite variable. And so once you use that information, you could kind of explore, do I need all of this training data to train a good model? Like, could I have skipped the low distance library, or could I have skipped the high distance library? Well, it turns out you do need both, but first I'll show you why. So if you look here, what I'm showing you is the LSTM, which is the RNN, and the RF, and the accuracy score. So if you train the model on only the 2T library, so that's here, with data that came from here, and you try to predict binding and non-binding for either low distance variants. So these are variants that are now less than 5 or less from the original wild type RBD. You can see it's actually really good. No problem predicting on low distance. But then when it starts predicting on high distance, it really falls apart. The random forest falls apart for both predictions, both low and high. Similar results can be observed for antibody escape. You can see that the high distance cannot be covered by the low distance training data. Now, you could look at the high distance libraries here, and they seem to be sufficient to cover all of the ACE2 binding accuracy, very high accuracies here. So you might argue you don't even need low distance libraries to predict ACE2 binding or non-binding. You could just get away with the high distance. That turns out to be potentially true for ACE2, but it's not true for antibody escape. And what you see here is there's not a particularly good performance for predicting binding of antibodies when you at the low distance space when you use a high distance library. So why is that? Let's unpack that for a second. Basically, it does not have enough training data samples of low distance. So every time it sees a low distance variant, it can't accurately classify it. So believe it or not, you actually need sequences that look really similar to wild type that are both binding and non-binding in order to more effectively train the models. So this was one of the most important and key innovations that if you are not really deeply entrenched, could miss. And you could end up with essentially very ineffective or subpar machine learning models. So really understanding the mutagenesis space was key to this. Okay, so what happens when you use all of the training data, low and high distance? The results are astounding. So of course, ROC curves, which are basically a way of plotting true positives to false positives, where you use the area under the curve as a metric, is one really common way of assessing the quality of a machine learning model. And what you're looking at here is that the closer you are to the blue line, the better the model is. The closer you are to diagonal, essentially the model is not particularly effective at all. It's really hard to see here, but what's plotted on the diagonal is essentially the random forest or the RNNs when the labels are shuffled. And what ends up coming out in the lines that are actually plotted towards the left-hand side of the graph is actually the models when they're trained with the full training data set. And you can see these areas under the curve are really strong values. So the closer to 1, the better your model is. And you can see all across the board for ACE2 binding. Sorry, this is ACE2 binding. I forgot the labels here. The red is ACE2 binding, and these are the 4 antibodies, which are missing the labels, but they're essentially Lycove, 16, 555, Regeneron antibodies on the right. And you could see that the area under the curves are all fantastic. And even the accuracies at low and high distance are all very high now. You know, everything is approaching 90 to 95%. So that's really the key to, and really the key to this was understanding your training data and making sure that you covered the mutational space. So what that means now is we have extremely accurate machine learning models that can predict on combinatorial mutations to the SARS coronavirus 2 RBD sequence. So what can we do with this? Now the fun begins. So first, I just want to note there's some key differences when you compare deep mutational scanning to deep mutational learning. So of course, we talked about this really prolific work by Jesse Bloom's group. And so one of the ways that they often analyze the data is they do something called escape maps, where they essentially look at the enrichment of an escape of sequencing data before and after the escape selection. And so that's how they come up with these, logos where they indicate potential mutations that escape. And these are the 2 Regeneron antibodies pictured here. Well, we can actually do something pretty similar with deep mutational learning. We can extract, if we use the random forest as an example, we can extract features that, that drive classification to, in this case, antibody escape. And what I'm showing you here is those features when they've been extracted, plotted. Essentially, the features are individual positions and mutations to certain residues of the certain residues, right? And so if you take a look here at Regeneron 10987, a really interesting observation here is that you see a lot of escape. Wait, I think I've made a mistake. I apologize. This is actually escape is red in this version and binding is blue. So please forgive me, but escape is actually red. And so what's really interesting here is that there are mutations in position 503, 504, and 505 that indicate escape from Regeneron 10987. That mutation is not observed in the deep mutational scanning data from Jesse Bloom's work. So that's an interesting feature that may be more powerful. We of course have to experimentally validate some of these, but I'll get to that in a minute. The same can be done for all of the antibodies. And you can see as an example here, is it Lycove 555? Yeah, E484 is obviously indicated here. In mutations, not just E484K, but other mutations in E484 identify. E484Q is, I believe, the kappa variant, and you can see it's being pulled out here. So this is one aspect. So can do most of the things that DMS does, even though we didn't train it, we didn't use single mutation data. But the other thing it can do is essentially you can compare it with combinatorial mutations. So if you take a look, remember that the escape map here from DMS doesn't actually come up with mutations in 490, and it doesn't really show you strongly the E484K. Well, if I plugged in these 2 mutations into both the random forest and RNN models, these high values indicate that these mutations do not escape Regeneron 10933. So that's good. Actually, that's completely consistent with the data from the DMS, okay? But here's where it's very powerful. You can actually combine these 2 mutations and you show escape. And that's something you could never do with DMS. And so this is where the power of deep mutational learning is the fact that you can start to combine mutations. And remember, if you can accurately, if these models are truly accurate, you could predict on what is our theoretical sequence space of the region of 10 to the 12. No experimental approach would ever allow you to explore 10 to the 12 variants, but we can now do it quite efficiently. So just to show you a couple more examples, because I'm probably running out of time, but I think this is kind of interesting and worth looking at, is we started, we have 10 to the 12 sequences we could explore. So we decided, well, so many of them are irrelevant So instead, let's focus in on variants that could potentially emerge because they're following some sort of lineage. And so we created synthetic lineages. So essentially, we took variants. This would be the alpha variant, right? And this is similar to the beta variant, and this one is similar to the kappa variant. And we created synthetic lineages to identify additional mutations. So this is the distance. So this is now 3 mutations from wild type. This is 5. This is 7. And we made predictions using the machine learning models on whether these additional mutations in these lineages would lead to ACE2 binding or non-binding. It's a bit of a spoiler alert, but of these 46 sequences, we were correct 43 out of the 46. And how did we confirm it? We experimentally expressed all 46 sequences. We did all the flow cytometry. And what you essentially see here is then reflected here that 43 out of our 46 predictions were accurate for ACE2 binding. Also importantly, of the antibodies that were predicted to bind ACE2, we of course also then made predictions on antibody escape. And that was then all of those 46 variants were labeled with all of the 4 antibodies and then experimentally assessed. This is essentially flow cytometry data of the mean fluorescence index on the x-axis and the model score, so the probability score. So escape is towards the left-hand corner here. And essentially, the escape prediction accuracy was, again, around 93% across all these 4 antibodies. So we're hitting that number really well. So the data from the test data accuracies line up pretty well with these synthetic lineages. And so it really shows you we have some pretty powerful models, and we can really predict these combinatorial variants. Notably, if you consider that we're here, this is basically the beta variant, or 2 mutations is even the delta variant in terms of the number of mutations from wild type. Only 2 more mutations from here, or actually 1 more mutation from beta, but 2 more mutations from alpha, and 3 mutations from the original SARS-CoV-2 will actually result in an antibody, or in 3 different variants. And this is only 2 nucleotides away from alpha, but 3 different variants that would escape all 4 clinical antibodies. And this is just the mean fluorescence index information, and showing you this is the wild type binding, and this shows you they basically lost almost all antibody binding. Why is that important? These 4 antibodies, of course, if these variants emerge, they couldn't be treated with these drugs. But more importantly, again, remember these antibodies may be reflective of human vaccine-induced immunity to some extent. So such a variant would certainly be of some potential risk for breakthrough infections and resistance to vaccines. Okay, and then one more fun thing that we can do. Of course, most of you, or many of you, have heard about this revolutionary advance by Google DeepMind called AlphaFold, which is a protein structure database, or 3D structural database they've created, where you can basically type sequences in, and they've used a whole lot of machine learning on structural predictions to try to predict possible 3D structures of proteins using sequence alone. So we did that. We plugged 8 synthetic variants in that are from here. I think that's from the beta variant lineage. And in all of these variants were of the ones we've experimentally validated, were correct in terms of ACE2 binding. And that's what's indicated in green or red. Green is ACE2 binding, red is non-binding. Here's the original SARS-CoV-2. And what's actually kind of notable here is if you look at the red, these are the ACE2 non-binders. They look pretty similar to the wild type, but there's a whole lot of ACE2 binders in green that have a completely different predicted structure than the wild type. That's a little bit scary, actually, because it basically shows you the extent to which SARS-CoV-2 can remodel and undergo mutations to this receptor binding domain and still maintain binding to ACE2. And presumably, those structural conformations, though, may lead to antibody escape. All right, very last couple of slides. Just bear with me, because I think this is really the punchline. So you may know that Stanford has put together a really nice database where they actually do the susceptibility index of SARS-CoV-2 variants. And so what they're doing here is all of these are the clinical antibodies, and unfortunately they use different names. But for example, BAM is LICO 555. And they've shown you that they've curated experimental data, and they basically show you almost every variant, of course, is escaping this antibody. And so they've amassed this data retrospectively, after all of these variants were emerged, they realized they're important, and then a bunch of experiments were done. And so obviously with a global pandemic and eventually an endemic virus, there will constantly be new variants that emerge. They get deposited online, and it's a long time before we know if they're any consequence. Because usually to know if they're of consequence, you either see it spreading a lot in a population, or you have to do all of these, very elaborate experiments. Well, the power of deep mutational learning is you can actually just plug in any variant and any sequence in the sequence space and get a prediction right away on escape from these given antibodies. And so if you look here, this is actually the alpha variant. And we took every ACE2 binding sequence that is one distance or one mutation away from alpha, and then we just looked at our two models. So each heat map here is the RNN or RF model for each of the 4 antibodies. And blue means escape in this case. And so what you see here is that, as we know, here's some escape variants from alpha, 1 mutation away, and then here's 2 mutations away. Well, you can start to run this on other variants like beta and kappa, and you start to see some pretty consequential things. Most notably, take a look at here. Here is a variant that is 2 mutations away from beta that escapes all 4 clinical antibodies. You get this profile, and why this profile could be really powerful is you could essentially see a new variant and then know whether a particular drug also is useful. Clearly, if you get 2 mutations away from beta, most of these antibodies, most of these variants would render these antibodies ineffective treatments. So this is a really powerful way to get an immediate prediction of a consequence. Of course, experimental studies should be done as follow-ups, but that can take time, of course. The other thing you can do is, instead of escape maps, we can build what are called escape networks. And this is a very complicated plot, but I'll try to explain it really quickly. So this is the alpha variant, and every dot inside the circle represents 1 mutation away from alpha, and every dot outside represents 2 mutations from alpha. And essentially the colors of these larger circles indicate escape from either 0, 1, 2, 3, or 4 antibodies. And so you can see alpha, it takes a lot, or there's very low likelihood that there'll be a variant that can escape all 4 clinical antibodies. But as we run through beta and kappa, you start to see there isn't as much for all 4. There's a little bit of an increase for 4 for beta, but there's quite a bit that escape 2 or 3 antibodies now. So this is another really informative way to rapidly understand kind of the sequence space and breadth of a given antibody or antibody therapy. And then it comes back to my first slide. So how can we, apply this? How can it be powerful? So if you can see that this study, or this headline, was from just about a little over a month ago, and it described this new variant, B11523. So because we don't have to experimentally express B11523, we could just literally go in and look up the sequence. We looked it up, and it has these 2 mutations in the RBD, E484K and S494P. And deep mutational learning predicts that it escapes Li555. And this is every 1 distance and 2 distance variant. But then you can look again at other distances, including 2, and you start to see that if this variant started to spread, it would be consequential potentially, because there's quite a few variants that would escape all 3 antibodies. 3 of the 4 antibodies, some that might escape all 4. And then, of course, these same kind of plots can be made. And again, the point is the information on this sequence was reported a month ago, and we could just automatically or very quickly generate this data and do this analysis. Good. So I'm just wrapping up now. And just to summarize, I think that I've hopefully showed you that deep mutational learning is a really powerful way to apply machine learning guided protein engineering applied to the SARS-CoV-2 context. we can identify a highly diverse landscape of SARS-CoV-2 variants that maintain binding to ACE2. That's sort of an indication that there's a lot of room for the virus to continually mutate. So that is certainly of concern as we go forward. It also allows us to do this prospective profiling of antibody escape. So essentially getting really fast answers to whether any variant has an impact on escape from antibodies. And I think that this, for the future, could be used to guide the design of combination antibodies therapies. So if we make new antibody therapies in the future, if those become necessary for COVID-19, you can try to mix and match antibodies that cover, the full scope of mutational coverage, so that you can have a less likelihood of an escape. And then I think really, kind of futuristic is that it may be possible that if you apply this approach on panels of antibodies, they could be, a huge mix of monoclonal antibodies, or it could be, polyclonal antibodies derived from, say, vaccinated serum. You may start to be able to predict variants that have a higher chance for vaccine resistance, and essentially higher likelihood. So maybe you'd have a chance of creating a scenario where you can rank order certain variants that would be a problem if they ever emerged. And of course, if they ever do emerge, then you may have a good idea already that travel location needs to be secured, or that place has to undergo more scrutiny. And I think that's what the really powerful opportunity may be in terms of the application of this technology. With that, I want to thank really an outstanding group of researchers. So this was a collaboration between my academic research group at ETH Zurich, and then also Cedric Weaver, who is a member of a spin-off company, Deep CDR, who worked very, very closely as well on this project. Joe, Roy, Lester, Sean, Jiami, and Beichen were all outstanding in just taking this project and moving it forward. We literally started it in March. We hadn't, obviously, variants we didn't think were such a big deal until then. And then we made a lot of progress in just about a little over 6 months. So I'm extremely happy with their performance and their productivity. Also thank all my members of my group who may not have contributed directly to this study, but are doing, of course, other very interesting things. So with that, I apologize for going a little bit over time, and I hope that there's still an opportunity for 1 or 2 questions if there are any.
Thanks so much, Sai, for this super interesting talk. So do we have questions from the audience? You can raise your hand or write in the chat. If not, I will just start with one question. So yeah, there is one question in the chat. Is the dataset available?
It's not available yet because we haven't published yet. We haven't even submitted it yet, but we will certainly make it available once we get to that stage.
Yeah. And so one question I have to the machine learning analysis, you showed that several machine learning approaches were had quite high accuracy, above 85% or something like that. And some of them were quite basic, like the logistic regression, for example. Do we understand why even very simple machine learning approaches work so well on this problem?
I think that one of the reasons is really just that, those machine learning models, the basic ones, were trained with the full training data set after a pretty careful analysis of quality filtering and balancing. I didn't really talk about that, but it's important to mention. And so I think that it's really because the training data is high quality. Clearly, we've done an extensive amount of flow cytometry and sorting. I showed you kind of like the first plots, but if anybody's done display, like protein display and sorting, we did 3 or 4 rounds of sorting so that the populations that actually get sequenced are highly pure. And so essentially, you limit the amount of noise in your data sets. And so I think that, my simple answer is if you create a very, very good high quality training data set, you understand, the type of data you need to train this type of model for this classifier, then very simple models are extremely powerful. Yeah, so I don't think you actually need to have that sophisticated of a model, which, you know, in a lot of ways, a random forest is a pretty straightforward approach.
Yeah. Then Ryan has a question in the chat. What's the name of the circle plot?
I don't have a name for it. One of my very brilliant first-year grad students, Jiami, developed this, and I think we haven't named it in any way at this point. I guess, oh, sorry, we do have a name, actually. We call it a deep escape network, but I don't know if that's certainly not an official name for it, but that's what we're calling it for now.